Hello, everyone. Welcome to this installment of the iReceptor seminar series with two exciting speakers today. We have Jenny Jiang from the University of Pennsylvania, and we have Maxim Zaslavsky from Stanford University as the second speaker. So the first speaker is Jenny. She's an associate professor of bioengineering at the University of Pennsylvania. And her lab uses high-throughput sequencing and single-cell analysis in combination with quantitative analysis to answer questions such as how does the immune system develop over age, what are the molecular signatures in autoimmune disease and cancer, and all of this with the longer aim of developing biomarkers for diagnosis and therapy. And she was also, and her lab also developed, for example, a high-throughput technology for profiling antigen-specific TCRs. And I think this is also something that she is going to talk about today. So with that, Jenny, the floor is yours. Thank you very much for being here with us today.
Thank you, Victor, for such a nice introduction, and also thank you for organizing that with Go and others. And so today, I'd like to talk to you about some of the technology development that in my lab on high throughput and high dimensional single cell analysis. And so hopefully I will have time to cover sort of three different areas that we have been developing technologies in. So I think this audience really appreciate the complexity of the immune system. And so decades of work that we have led us to really have an understanding of, in many of the cellular and molecular event associated with different immune responses down to the tissue level. And however, I think that one of the questions is the composition of T cells and the B cell receptors, which actually bear the sensitivity and specificity and also long-term memory that allow humans to respond to infections and vaccination, but also remember so that when the next pathogens come through. And so these characteristics have taken advantage by humans as a form of vaccine for centuries. And now their ability to actually fight against cancer has been realized. So the reason that the T cells and B cell has ability to recognize such a large range of antigen lies in their fundamentally how they generate their receptor diversity. So with using the VDJ combination, and so let me use a pointer. So using a VDJ combination, the human genomes can use relatively small fixed number of gene segment to generate a large diversity of receptors. And unique to B cells, when the antigen encounter, it will actually go through another round of diversification called a somatic hypermutation to further diversify the antibody repertoire. Where for the T cell, once the T cell receptor is generated through this VDJ recombination and it's not changed, so it's become an internal ID for T cell proliferation and clonal expansion. So this is a process really like an automatic tool where you have a fixed handle, but then you can continue switch the head to match different antigen. So the potential immune receptor repertoire diversity lies in anywhere 10 to the 18th or even larger. However, at any given time, each individual humans are actually express a much smaller set of what is possible. But nevertheless, diversity is the birthmark of B cells and T cells. So when the B cells and T cells are just recombined or generated, they're naive. And the cells are smaller and compared to activated cells, and they're actually morphologically look similar. But uniquely, they each of T cells and B cells express a unique antigen receptor, and the purpose of them is to find and match the antigen. So under the stimulation of immunological disease or vaccine, those cells undergo drastic changes. So both morphologically as well as functionally and phenotypically. So how to understand, cells go from point A to point B, what are the changes they make, how to make those changes are essential to understanding many human diseases and for therapeutic development. So that's where my lab is, and we're studying the systems immunology, where we develop high throughput technology in combination of informatic analysis and modeling. And we use this approach to directly study human disease in human. And actually, this approach actually allows us to answer some of the mechanistic questions in human directly, which will lead to biomarker discovery. And then we can also further take them to personalized early disease detection and the immune engineering of therapeutics. So today I'd like to talk to you about the technology development in 3 areas. And the first one is in the bulk repertoire sequencing. So forego of the single cell pairing, but we focus on one of the chains of T cell receptor and the antibody, and to have a large a diversity survey. And the second one is in the area of high throughput T cell receptor affinity quantification, which we're also very interested to develop into many of the therapeutic development based on this. And the third one is our new technology development in the multidimensional single T cell profiling. And so the first one actually started when I was a postdoc in Steve Quick's lab decades ago. And so using basically the first generation of high throughput sequencing 454 at the time, and which can measure about 400 base pair, we are actually using zebrafish as a model system to analyze that. So I couldn't find a fish picture that illustrating our concept of, getting the fish smoothie and then to getting the RNAs and then gene-specific primers. But this is actually an artwork, I think, displayed in one of the museums in Germany. So then after the 454 run, you have 1 million sequence generated overnight, and you can do all the different informatic analysis. And through our initial analysis, we did, the antibody heavy chain in fish, and depending on different way of estimate, there are several thousands heavy chain that fish express. But those early studies led us to quickly realizing there's, many caveats in the first generation of immune repertoire sequencing. For example, high throughput sequencing era and also the PCR era can generate artificial diversity. And also they give an amplification bias. So for example, things that you put in the RNA is 1-to-1 ratio, but the sequencing rate is not. So that's generated altered clone size. And also there's a low coverage of the diversity, then you're not getting, all the sample information. And there's some problem with, different cell number inputs. For example, some protocol is good to analyze small amount of cells, where other is large number of cells. And so we want to develop something sort of addressing those questions. So we used a unique barcode, so MID, which is also used in nowadays single cell RNA sequencing. So by fusing this MID with the reverse transcription-based primers, and then when we can generate the cDNA, the barcode is basically transferred. And so this information carries through all the PCR amplification process. So during a data analysis, you can, in the first level, you can look at a barcode based on the MID barcode. And for example, those are all the MID barcode number 1. But then we quickly realized that different T cell receptor sequences might actually accidentally tag with the same MID. But because their VDJ is so distinct, and coarse level of clustering can separate them. So after you group them into subclusters, and you can build a consensus to remove those errors associated with sequencing and the PCR. So that gives actually an accurate sequencing read of individual receptors, but also give you a counting of the receptor abundance. And so effectively, we improve the sequencing accuracy about 150 times. And so how we also like to validate this in a varying number of cell input. So we sorted naive B cells from anywhere from 1,000 to 1 million following this protocol. And we basically sequencing them at different depths. As you can see, about 5 times of the cell input essentially reaches saturation. However, if you don't do this kind of barcode, so as you sequence and you actually discover more diversity. But as you know, for 1 million B cells, even both chromosomes recombined, you maximally have 2 million diversity. And so many of those are just artifacts associated with sequencing and the PCR. So if we take the saturation curve for different cell amount and then plot the correlation, this actually the slope gives us the repertoire diversity coverage. So we're discovering more than 80% of the sequence in a sample. So using this, we actually did several studies in the antibody where we're looking at young children's, how their repertoire responds to malaria and in HIVs, and how B cells, mutate actually under the pressure of the virus. And we also developed a similar technology to sequence the T cell receptor. In this paper, we actually documented, different way that you can QC this method. And we also used it to looking at the TFH, which is a very important T cell subset in generating antibodies and also as a reservoir for HIV infections. And so those are published, and so I won't talk too much. And so this is I want to just say that although here now people are focused on single cell technologies, but this is still remain a main tool that you can actually do a lot of the cells at the same time. So we continue using them in our large cohort studies. So second technology I want to talk to you about is the iTAST, and then for this is an in situ T cell adhesion based affinity test. And so, and for T cells and for CD8, and they have T cell receptor on the T cells and CD8, and they bind to the pMHC molecules on the antigen presenting cells. So previously we have developed a way of quantify the two-dimensional T cell receptor affinity, because this is a surface interaction, so it's two-dimensional compared to, for example, flow cytometry or surface plasma resonance, which is called a three-dimensional T cell receptor affinity. So previous studies have shown that a two-dimensional T cell receptor affinity actually correlated much better with T cell function. However, the challenge is that you do that and require either transgenic mice that entire mouse express only one T cell receptor, or you have to previously, for example, expand the cell so you have more copies so that you can repeat the assay. But in the assays, so it's rely on the adhesion. So here the movie actually cycles between two adhesion events. One has an adhesion and this one does not. So the one has an adhesion, you can see that there's a little blimp of the red blood cell membrane. And so that actually mediated by a single T cell receptor on the T cell side and interacting with single molecule, MHC molecule on the red blood cell side. So if you're changing the contact time, the adhesion probability will increase. So anything that changing with time, you can actually extrapolate the interaction kinetics, and the saturation, the plateau state actually give you the affinity. So this is how we actually estimate the interaction, the T cell receptor affinity. And so what we did is to basically develop a method to actually measure primary single T cells from human samples. So to do that, we actually take a dissociable tetramer called a streptomer to enrich and isolate antigen-specific T cells from human samples. And then we subject them to large excess amount of biotin to dissociate off the streptomer, and then leave a clean T cells to be tested in the adhesion chamber. And after the test, the T cell is transferred into a PCR tube to amplify both alpha beta paired TCR sequences. And so then this you can relate it to then affinity of the T cell receptor. So this is still single cell manipulation. So in a day, we can do actually about 70, 80 cells, but that's already orders of magnitude higher than vehicle because that's limited by protein purification and others. And so here we're using this and start looking at the antigen-specific repertoire in humans. And here we focus on one particular antigen, is HCV, from the HCV virus. For healthy individuals, the blood donors, and they're routinely screen them for HCV infection. So those individuals are negative. However, you still have the HCV-specific T cell receptor that prevent you from, or help you for future possible infections. So we use enrichment and to looking at each individual's HCV specific T cell versus polyclonal T cell. We looked at 9 individuals, and some individual actually came back for multiple donations. So we have multiple blood draws from them. And so we can compare the median for the affinity range. So surprisingly, we actually noticed that there's about a 1,000-fold difference in affinity for this polyclonal antigen-specific T cells. And if I put in, for example, the affinity for a mouse high affinity TCR OT1, and you can see that in humans, there are many T cells that has much higher affinity compared to OT1. And so we also noticed that there's this interesting trend that younger individuals tend to have higher affinity compared to older individuals. So that may explain why older individuals are prone to infection, but also less responsive to vaccination. So why high affinity TCRs are attractive? Because the higher the affinity and the fewer antigen that it requires for T cells to recognize and then generate effective adhesion toward them. And so hopefully I convince you that this TCR affinity is part of the T cell repertoire profiling metrics. And maybe that some patient who undergo immune checkpoint blockade that failed it may actually require additional therapy, for example, T cell receptor redirected adoptive cell transfer therapy to compensate it, because if you can think, if they don't have the high affinity TCR, then maybe no matter how much the break you take from the T cell, they won't be effectively capture the cancer cell. And as I mentioned, we can get a T cell receptor sequence. So we can actually transduce the T cell receptor sequences to either primary cells or some T cell line. In this case, Jurkat does not have the endogenous TCR. And then we can also validate the T cell receptor affinity as well as test the function. And just to highlight why this method is maybe important for adoptive cell transfer therapy, and we analyzed some of the published data on single cell RNA sequencing of cancer cells. And for both neo-antigen as well as cancer testis antigen, as you can see from those 2 patients, there's a large degree of heterogeneity. So for current strategy of selecting one antigen and finding that antigen-specific T cell receptor may not be enough. So we think that maybe looking at antigen heterogeneity and then in combination of selecting a combinatorial T cell receptor cover all the different antigen heterogeneity may actually improve the personalized therapy. So I want to spend the rest of the time to talk about a new technology we developed called TCR-pMHC or TCR-pMHC HD. So actually the name stands for tetramer associated TCR sequencing or tetramer associated TCR sequencing high definition. And so this is where we actually integrated multiple dimensions of information onto single T cell. So as I mentioned, for someone interested in T cell, I think these 4 dimensions of information are really critical. What is the T cell receptor sequences? And what antigen specificity does those T cell receptors recognize? And what are the function of the T cells and the phenotype? And so when we start this and we ask, maybe we can actually link, two information and how to link the T cell receptor sequences to antigen. And so this question has actually been extensively studied in the field, started with Mark Davis's development of tetramer, and in its original form, and it only can detect parallel, basically a few antigen at the same time because of the limitation of the fluorescence channel in the flow cytometry. However, at the New Wells in Mark Davis lab and also at the same time Tom Schumacher's lab, and developed this really clever way of expanding the number of fluorescence channels by using a combination. For example, the green, red, blue, each individually encoded an antigen specificity, but in combination, they can encode additional antigen specificity. So with this, you can increase the number of antigen to about 15 or more. And so what's more than this is using iron isotope. So they're using cytol. And so using this mass cytometry, you can label the MHC molecules now using mass isotope now is expanding the number of antigen to over 100. And so we thought, what's more than that? And the answer lies in the DNA. So if we're using DNA as a barcode, essentially there's no limit. So we can actually continue increase the number of base pair to accommodating a large library. But now we're routinely doing more than 300 epitopes in the experiment. And so traditionally, when you actually generate a peptide-C, and you actually need to chemically synthesize the peptide and then fold it with the MHC molecule. This takes several weeks, if not months. And so Tom Schumach's group came up with this UV exchange method, actually significantly speed up the process. So when you fold the MHC molecule, you can give them a placeholder peptide that is sensitive to UV. So when you shine UV, the placeholder falls apart. And if you have the rescue peptide in the solution, then the rescue can take place and to become a new species. So in a few hours now, you can actually easily generate a large panel of peptide-MHC tetramer. So now the bottleneck is how can we generate a large panel of peptide and quickly and more affordably. So the way that we solve this problem is by synthesizing DNA oligo that encode the peptide. So then part of those oligo, then we can use them to generate a peptide using in vitro transcription and translation, and then UV exchange it to MHC monomer. So the other part of the DNA oligos, and we can become the unique DNA barcode for antigen specificity. So then we can just, putting a common handle onto streptavidin and then a neo to individual DNA oligo. So then when you generate a tetramer by putting, monomeric MHC with the streptavidin, now you have plateful, 96, 384, individually labeled DNA barcoded peptide-C, and then you can also label them with very few fluorescent fluorophores and can help in sorting. So this effectively lower the cost about 20 times. And has a turnaround time about 2 days. Half of them are cancer or endogenous type 1 diabetes related peptide. The other half are foreign antigen. And so, and we can sort, for example, the single specificity and then amplify both the T cell receptor alpha beta and then also the peptide-C barcode. And so we thought about using this to looking at, neo-antigen. Because the neo-antigen in most part is a single mutation from the wild type counterpart. And however, previous study have shown that if there's a significant cross-reactivity that is happening, then in the mild case, that leads to skin color discolorations, but also in the severe case, it could lead to blindness or even death. And so we want to understand whether there's T cells that can recognize both the neo-antigen and the endogenous, and if there are ways to tell them apart. So we curated about 20 pair neo-antigen and wild type counterpart antigen from the literature, and we label the tetramers either neo-specific or wild type specific. And so then based on this true fluorophore, you can also sort the third population, which is cross-reactive. So here, if you're looking at the single specific cells and apply the background noise threshold, and most of the cells indeed recognize single species, whether it's neo-positive or wild type specific. But if you're looking at the gate that's sorted from the cross-reactive cells, and after the threshold, most cells recognize 2, and some of them actually recognize more than 2 different antigens. So now we have a set of antigens as well as T cells that either specific or cross-react. So now we can look at the pattern on the neo-antigen and to basically evaluate if there's any characteristic of how likely that they can generate cross-reactive T cells. And so we arrange the neo-antigen based on their position. So the peptide is about 9 to 10 amino acid. Some of the mutation lies in position 3 and 4, many in 5 and 6, and a few of them in 8 and 9. And as you can see from this figure, And if the neo-antigen is at the both end of the peptide, and then they have actually tend to induce a lot more cross-reactive T cells compared to if the mutation is in the middle of the peptide. And so this is also statistically significant. So this suggests that the neo-antigen with mutation near the fringe elicits more cross-reactive T cell responses. So this finding actually is consistent with Mark Jenkins' study where they used the alanine scanning to looking at the peptide cross-reactivity in the context of CD4 cells. And they found the same thing where the alanine scan in the both end regions actually induce more cross-reactivity compared to the middle. So the next question is, how can we actually, integrate all 4 different dimensions onto single T cells? And so to do that, we combine our DNA-labeled tetramer with DNA-labeled antibody and then use them to stain CD8 T cells. And after cell sorting, the cells are loaded to a BD Rhapsody platform, single cell amplification, to specifically amplify the DNA barcode for antigen specificity, the T cell receptor sequences, gene expression, and phenotype markers for the antibody. So with a 20,000 cells per experiment, and now we can actually do that all 300 and more tetramer all in one experiment. So what TCR-pMHC can do is to distinguish, for example, disease groups using known antigen and also screen for new T cell antigen, validate antigen expression, identify therapeutic T cell receptor. Probably more interesting is to looking at, identify clonal expansion and activation status of antigen-specific T cells. And so we want to, really characterize all the different parameters and noise and robustness of the assay before moving to human samples. So here we use 6 different peptides and to sort and stimulate 6 pools of polyclonal T cells. And then we grow them in large number. So then some of them we can use the bulk repertoire sequencing to understand the T cell receptor composition. And then the rest, we subject them to TCR-pMHC. And so as you can see, all 6 peptides has identified cells. And we can also using the bulk sequence generated T cell receptor to actually serve as a gold standard of what's in it, to actually looking at the recall and precision. So except GAD have a little lower recall, everything else was really high in both recall and precision. But most important is the false discovery rate is less than 5%. So we want to understand why a little bit more, why GAD actually generated such a low recall. So we zoomed in GAD, and actually there's one T cell and take over in the culture. However, they did not bind to our antigen. So this is also suggests that in the more quantitative DNA barcode counting actually is more sensitive and detecting, the cross-react or the bystander cells. So with this, and we're moving to human cohort studies, and here we curated 10 healthy individual and 8 type 1 diabetes patient, and we have 215 about type 1 diabetes related antigen, and about 100 or so, viral and other foreign antigen. And we use them to screen the CD8 T cells and then isolate it from this subject. So we actually have 35,000 cells and accumulated from 4 different chip runs. And as you can see, they relatively have fewer batch effect. And of those cells, and they actually form 13 cluster. And then cluster 13 is our spiking HCV specific cells. And that's very distinct. The cultured in vitro expanded cells are very distinct from the primary cells. And then cluster 1 to 4 are both naive, and cluster 5 is a NK-like T cells. Cluster 6 is a central memory. Cluster 7 is transitional, and 8, 9, 10 is effector memory, and 11, 12 are effector or TAMRA population. So as I mentioned, we also have about 70 different antibodies, And so, for example, here are listed several canonic markers, and CD45RA and RO are two splice variant of the same protein. And if you do RNA sequencing, unless you do a full length, it's really hard to distinguish these two. But if you, of course, if you use antibodies, it's very easy to tell them apart. We also have the T cell receptor data to guide us to look at the clonal expansion, consistent with the clustering and phenotypes. And so the naive cells are mostly absent of clonal expansion, but then the memory and the effector cell population have very degree of clonal expansion. Of the 300 or so antigens, about 15, the top 15 we listed here, and most of them are foreign antigens. They are residing in non-naive, and Marta antigen is something that is self-antigen. It has been documented that healthy individual actually has a very high frequency of T cells that recognize T cell identified. And as you can see, there's a large degree of heterogeneity in terms of their clonal expansion. And so this is probably reflected from, on their vaccination and also infection history. However, we did notice some antigen-specific cell phenotype. For example, the influenza epitope M1, all the T cells that recognize it, it's in the transitional cells, and the EBV has two different stages. One is latent, one is lytic, and the protein associated with the latent phase induced T cells in really clustered in the central memory, where the lytic phase protein has T cells that recognize it in the memory, in the effector memory and the effector cells. So this is also consistent with a renewal study that uses situ. So of the 200 or so type 1 diabetes related antigen, we identified 3. Their T cell, counted T cell frequency actually differentiated from healthy from the type 1 diabetes patient. And so this is really interesting. It could maybe serve as a way to perform disease diagnostic. But also I want to caution everyone that this is a cohort of adult patient. So it will be interesting to looking at in pediatric patient where it's early disease onset and see if the same antigen or maybe a different set of antigen could actually be the disease marker to look at it. So in a study, we can also identify T cells and transduce them to primary cells or cell lines. So here we identified 5 different T cell receptor and 2 for EBV and 2 for 2 different type of type 1 diabetes antigen. And another one is really interesting where this T cell receptor actually cross-react to 3 different antigen. One is the frame-shifted insulin, and the PTPRN is IA2 protein, which is a known type 1 diabetes related antigen. And the third one is actually from a microbiome component that is very common in gut microbiome. So it's interesting to see that the T cell receptor can actually cross-react to 3 different type of antigen. So here is a tetramer staining, and then we also did a functional analysis looking at a CD107 alpha expression, and 2 different ways to validate this. We also recently made available of the technology for CD4 T cells, and then looking at various bacterial peptides and in healthy individual. And to our surprise, we find, at least in this cohort of healthy, has a large fraction of cells that recognize Mycobacteria TB. So this was alarming, and we thought, why this large TB related T cells. And then later on, by doing sequence search, we found this TB antigen actually has exactly sequence as some of the commensal bacteria. So then we validated in the cohort study in bulk and using the tetramer to do the staining and then confirm that indeed, and those individuals are having this population, probably because the commensal bacteria is actually stimulated prior, clonal expanded those T cells. To conclude, so I've talked to you several different technology that we developed in the area of bulk sequencing, TCR affinity quantification, and the most recent one is integrated multidimensional single T cell profiling. And so TCR-seq HD provide an integrated T cell profiling on antigen specificity, TCR sequence, gene expression, and phenotyping. And although I showed you an example in how to use it in type 1 diabetes, but this really can be used in many different other diseases, for example, infection, cancer, and also vaccination. So with that, I'd like to thank all the people that did work. And so especially those students, they really behind those innovative ideas in the methodology development, and our funding support, and also collaborators. And thank you, and I'm happy to answer questions. And also I want to say, we have multiple postdoc positions available. So if you're interested, please reach out. Thank you.
Thanks so much, Jenny, for this fantastic talk. Do we have questions from the audience? You can either raise your hand or you can write it in the chat also. But we will monitor the chat. But I will start then with a question. So I think something that we talk a lot about in the iReceptor consortium is this problem of that each group has their own method of making their or generating their TCR data, and then it's very difficult to integrate all these data sets into one metadata set so that we can really profile, across many dimensions. What are your thoughts on overcoming this problem? Is it an experimental problem? Is it a computational problem? Because it seems like, we are still not at one, TCR sequencing method, one BCR sequencing approach. So how do you think?
Yeah, I think as we talk about, before the seminar started, and there's a lot of database to upload different things. I think that I think there's a need maybe to upload both raw sequencing data as well as annotated data. I mean, a lot of the annotated data, some people just listed using a table format in the supplementary information in the paper. I think unless those things are uploaded and put in a place where using a uniform format, otherwise it's really hard to compare, between different studies. And yeah, I agree. I mean, with a large number of data that basically accumulated, and also many of them associated with specificity, we can actually learn a lot. And that's one of the reasons that we want to develop this method, is to provide some kind of gold standard or reference. So as we improve on computational prediction and looking at whether there's way to predict, we can actually cross-check with experimental validated systems.
That's really nice. Yeah. Felix? We can hear you, Felix.
Yeah. I had a couple of questions, but just in terms of what Victor said, yeah, the AIR community is developing a single cell gene expression API, database model. So we'd be happy to talk to you about. So it seems like your data would, fit into that database model. As I get older, I get more and more obsessed with aging repertoires. So you mentioned
everybody should care about that.
Everybody should care. That's a good point. Yeah. But so you mentioned the decrease in antigen specific or high affinity clones, and you may have mentioned this, but is that just going along with the idea of lower diversity and fewer overall clones, or is it, do you think there's like a specific loss of antigens, high affinity clones, or is that even it's not possible to really discuss?
Yeah, so initially, so the data I showed you is in the peripheral blood. And so you could say, okay, well, maybe they're exiting the peripheral blood and they go to the spleen, but we actually look at the human spleen and they're not there either. So that raised a very interesting question. So they may be, they do, maybe either deleted or not very surviving, long-termly. So that could be a problem because at certain time, the thymic output is very limited. So then you don't have fresh T cells to support the pool, and they really live on whatever it generated before. So, yeah.
I guess I have a very simple kind of question. It's, if you just took a younger, healthier repertoire and randomly deleted clones, would you see this loss of high affinity clones, or is it even stronger than just kind of a random process? Could you ask that? I mean, I guess that's an
I think it's probably not a random.
Yeah. Okay. Yeah. Are there other questions, Victor? Because I have another question, but it's
we've looked.
Sorry.
Ask another question, Felix. Yeah.
Okay. Early in your talk, you talked about overall T cell receptor diversity and trying to estimate T cell receptor diversity. And there's so many estimates out there. And then there's estimation of total diversity in the human population. What's the potential T cell diversity? And you're highly sophisticated in mathematics and stuff. I was wondering, what is your, if you had to say, okay, a typical healthy human has how many different T cell clones? Just thought,
Yeah, I guess that's really limited.
We could turn off the recording, but yeah.
Yeah, but I think that's really limited by the sampling, right? Because pretty much what we can do now is to looking at the peripheral blood. Even peripheral blood, the analysis is limited of 10 million or a few tens of millions of T cells. So I think there's need to be sort of a consortium to looking at different tissues, organ. There's also a large amount of resident T cells and in different tissues and organs. So I think the diversity probably enormous as than what we have able to be appreciated. Yeah.
So, but those tens of millions, even in the peripheral blood, aren't going to be unique.
No, they're not. So, but you're still limited by the sampling.
Yeah. Okay. Okay. Thank you.
So, Jenny, one more question. So,
sampling size compared to
the connection is a bit bad, I think. Yeah. Jenny, can you hear us?
So sorry, your voice paused. I couldn't hear, or maybe on my.
Yeah, I think it's on your end. Yeah.
Okay. Yeah, I didn't catch the question.
Yeah. No. So the question is for the third part of your talk, where you look at the difference between type 1D and the healthy, and then you find these 3 epitopes. Could you elaborate a bit more what these epitopes are, and why do you think they could be important? I mean,
so those epitopes are previously discovered type 1 diabetes related antigen. It's not a neo-antigen, so we put a curated 200 or so type 1 diabetes related antigen. So there's a study that done in 2008 where they used only about 10 or 20 type 1 diabetes antigen. They did not find anything that can differentiate it between healthy and type 1 diabetes patients. So I think there's a need to put in a large number of antigens. But I think even more important is to looking into pediatric sample, because type 1 diabetes is an early onset disease. And so because the patient that we're looking at, they are already in their 40s and a few of them 50s. And so that may already pass the time where T cell clonal expansion and recognition of antigen is just onset to start it. So that may give us, when we look at a young pediatric sample, they may give us a different set of antigen.
And do you also pair your single cell sequencing with bulk sequencing to see where in the diversity spectrum the antigen-specific cells fall? Are they high frequency? Are they low frequency? Do you?
Yeah. So yeah, we did look at that. So most of them are not that high frequency.
Yeah.
So that's probably because one thing is they're peripheral blood, they're not tissue. Yeah. And another thing is that they are basically at adult and late, very late stage. So they've had these for decades probably.
So since they are since they're quite low in the blood, how do you? So wouldn't that be a problem for using then immune receptor data as a biomarker if the biomarker is so low in the?
Right. There would have been. This is to basically demonstrate, we can see things, but as I mentioned, this is a adult patient and then peripheral blood. Things might be closer to what's happening in tissue in pediatric peripheral blood. But then that may be more applicable.
But then how do you see the future for using immunoreceptor sequencing? So should one then always use tissue samples basically to diagnose and prognose a patient, or how do
I think if we have a good cohort of children pediatric samples, then that will allow us to confirm and even maybe peripheral blood and then compare with the pediatric tissue sample. Then we can do that comparison and see if there's similar antigens that are enriched both in the peripheral blood as well as the tissue.
Yeah.
And then those markers would actually be as diagnostic value. Yeah. And some of the T cells activation actually precedes the B cell. So I would think that they're more sensitive than some of the autoantibody. But even clinically, people have autoantibody and they may not develop into type 1 diabetes. So so there's need to be kind of a long-term monitoring and also looking at both peripheral and tissue to actually come with markers that way.
And so you worked on both in your career on both BCR and TCR, right? So but there are not so many works where people compare both BCR and TCR sequencing datasets at the same time. Is that also something that you're looking into, like comparing antigen-specific TCR and BCR sequencing datasets on the same disease? Is that something that you also work on?
Yeah. So we're also working on developing equivalent for the B cell, but I guess the challenge in B cell is that they recognize three-dimensional surface and have such a focused and a detailed epitope yet. So,
yeah. Great. Are there any more questions from the audience? No? Then thanks so much, Jenny, again for this beautiful talk. It was really nice having you here today.
Thank you. Thank you, Victor, and others for having me.
Thank you so much. And then we will wait for 10 minutes and then we come back at 5 p.m. Oslo time for the